welcome to some feedback for the Populations and Nervous System A2 test. So the first question, I appear to have a graph with time going on the bottom, days of the experiment, and population density. What's going on? We're growing two things, two parameciums, so PA and PC. PA and PC down there. And they're growing on a culture of a bacterium, Bacillus, which is their food. And they're measuring the population densities of these. For PA, on which day of the experiment did the population growth enter stationary phase? So this is the, if you remember the phases, lag, exponential, stationary, death. So when are we getting this stationary phase? So it's levelling off there, so we're having day 10 there. On which days of the experiment is the population PC, that's this one down here, in the death? So when is it going down? And it says in which days. So it starts going down at 2, ends, well, it's still going down right at the end. So we're going to have 2 to 14. The experiment shows both interspecific and intraspecific competition. What type of competition is most likely to have caused the population of PC to decrease after day 2? So these are both going up, then this one's going down, so it's more likely it's competing with this other one, PA, this other paramecium. So this is going to be our different species competition. So PC and PA, different species there. So that's inter, which means between. Intra means within. So we've got inter specific competition. What is the carrying capacity for PA in this experiment? Well, it's likely that the carrying capacity is the maximum population that the environment can support. So read off the scale there, and that's your answer. You need some units. You won't be telling me without the units, and the units, just copy what you got there. What a simple way of getting the units. So we're going to have 240 number per cm cubed. How might carrying capacity be increased? Well, the most obvious example is let's add more food. And what is the food? The food is this Bacillus. So go for the simple one, add more food for that Bacillus. Suggest with an explanation what would happen to the numbers of PC if PA became infected with a parasite. So essentially, if we got rid of PA, what would happen to PC? Well, PC would go up, presumably, because it's got all the food to itself. There's less competition for the food. So we're going to have increase, and here there's less competition from the other paramecium, so less competition for food from the other paramecium. Distinguish between, so tell us the differences between these two statements, density dependent, density independent. Well, the dependent one, so dependent, the effect on the population depends on the population size. So the effect depends, not caused by the population, but it depends on the population size. For example, food supply, like we've got in the example there. It doesn't want examples, we're just wanting these definitions. And independent, the effect will be there, and it doesn't matter what the population size is. So the effect is the same, whatever size of the population. So typical things like forest fires and things like that affect the entire forest. Obviously not in this example. Name one density independent factor that could have changed this experiment. So in this, where we've got a culture of microorganisms, we're going to have forest fires and floods and things like that. No. But something else that might affect cultures and affect all of them regardless of the size might be temperature or pH, something like that. So let's have temperature, or we could say pH, and we're definitely not going to say things like food and so on, because that would be the other dependent one. Question 3, we are just drawing an action potential now. So we're looking for, let's just sketch one out. We've got resting potential, and then this goes up and down, overshoot, and then back to resting potential again. There's one mark for drawing the shape, and then one mark for really getting the numbers right. So the shape, so we're going to start down here at kind of -65, and then we're going to go up pretty quick up to our plus 35 or 40 kind of value. Then we're going to go down again and repolarize, and then hyperpolarization, factory period, back down to our same resting potential again. And the time scale for this, I'm thinking about 3 milliseconds. Label your action potential to show depolarization, so we're going to have depolarization, you can write the full word there, and repolarization is this section here. Diagram below shows the structure of the membrane, so things that you recognise from in the membrane. From what type of biological molecules are pumps and channels? Those are proteins, you remember that from last year. And this bilayer, well, these are phospholipids. With reference to the diagram, so we're going to mention these things in the diagram. Describe how the following events are brought about. Resting potential, so we need to talk about the sodium-potassium pumps. So we're talking about sodium out, K plus in. And we need to explain how we get to this resting potential of a -65 value. Well, we could talk about, well, 3 sodiums out, 2 potassium in. That would create a difference in the two sides. Or maybe something like the permeability of the membrane to potassium is higher than for sodium, so more potassium would leak out than sodium, so more K plus leaks out than Na plus leaks in. So those would create this resting potential. In our depolarization, the impulse opens channels, so we're going to get the opening of Na plus channels, and the voltage gated channels, there. And what happens, these sodium ions rush in, so some idea of them coming in pretty quickly. So increased permeability to sodium via these sodium channels. Explain how impulses are transmitted across a synapse. Well, all sorts of things we could say here. We could say about the transmitter substance, or ACH, or acetylcholine, is made in the synaptic knob. The impulse makes these, if I draw one, I can't bother to write them anymore, the impulse is going to make these move to the presynaptic membrane. We're going to get calcium ions rushing in. The impulse is going to make the presynaptic membrane more permeable to calcium. We're going to get the vesicle fusing with this membrane and releasing the transmitter substance. We get diffusion across the synaptic cleft. So after exocytosis, we get diffusion of the transmitter substance. We've then got the postsynaptic membrane. We've got receptors, and so this transmitter substance will bind to receptors in the postsynaptic membrane. And hopefully in there we've hit 3 marks. I can't bother to write them, but you just need to look at your notes and put the right ones in. And then we have got an essay question, which is the nitrogen cycle. Just basically you need to grab your nitrogen cycle out of your notes and you'll be happy there. So don't forget to mention all the names of the bacteria, all the names of the processes, all the names of the different forms of nitrogen, where they are. The nitrogen, is it in the air, is it in the soil, is it in the root nodule, and get everything in the right order. So just look at your nitrogen cycle in your notes, repeat that for the essay question. Well done.